Okay. Let's start. Warm welcome to everyone in today's webinar. Um, I am from Jumla, Nepal. Our Shankar sir, keynote speaker for our today's webinar, he is from US. He is a PhD scholar. He already completed the comprehensive exam and preparing for thesis work. He have done enormous work on in silico kind of work, and he is currently rigorously working on QSAR, AI, machine learning, and some modern technique to incorporate in drug discovery. Hope it will be quite helpful for us. And this webinar is jointly organized by our research group and the Association of Faculties of Pharmacy of Nepal, AFPN. Uh, you can go through the Shankar sir's profile through the Google Scholar or somewhere. He have about more than 40 peer-reviewed publication, more than 500 citation, quite good H index and I10 index. Being an early career learner, his output is quite impressive and it's a positive asset for us to learn something from him. I would like to hand over today's stage to Mr. Shankar Thapa. Shankar Thapa, the floor is yours now. Please go ahead.
Thank you, sir. Thank you for warm welcome. So once again, I would like to welcome all the participants, and hopefully today's webinars will be fruitful in your research careers or academic careers. Yeah, so today we are going to learn about the QSAR and then like how QSAR helps in drug discovery. So that is the main motto of today's webinars. And then like, so initial 15 to 20 minutes I will take to explain what actually QSAR means and what is the theory behind it. Hope my sound is audible to everyone, right? So can anyone confirm it, sir?
Sir.
Okay, thank you. So yeah, so that's what like I plan to do today. So like initial 15 to 20 minutes I will take to explain the QSAR basic things and then what are the steps involved. Okay, so and then like afterwards we will go to the like practical sessions, like how it can be done by using open access software and then database. So my main aim is to make you familiar about the open access softwares and then database, like so that like it can be useful in like academic research, and especially for the students because we don't have fundings and then financial support to do the research in Nepal. So for that reason, that's what I always think. So whatever I am doing, so I try to do and then try to find out the open access resource so that it can be useful for everyone. Open access yet say like very authentic or like who can give the near to accurate result. So that's what like we're going to do today. Hopefully now I will share my screens and then like so that I will start the QSR sessions for the theory part first. And then why I am doing these things, okay, so like because QSR is quite vast and then complicated topic to address. Yeah, so what I come to the conclusion is if I go through direct to that all vast and then complicated complex so things, so it will be quite difficult to understand for the undergrad students. So today's sessions will be more focused towards the simple software, like from where we can get the result, authentic result, yet it's very a simple way. So within 2 to 3 clicks we can get all the results. So that's what I wanted to do in the webinar. So yeah, so that's what like we will do today. So that like tomorrow sessions we can do all the steps through comprehensive way or rigorous way. So each and every steps we will do that one like completely in-depth analysis in tomorrow's session. So today will be like superficial and surface. Superficial, it doesn't mean the results and then procedures is not reliable. Okay, so all the procedures and then the results of the today's sessions are also equally reliable and then equally authentic. But only the things in today's sessions, I'm going to use the software who that software is based on machine learning and then AI. So that's why like most of these steps are, automatically built in the software. So that's what we will do. Okay, so let me share my screen. Entire screen. I think like sir, any कसैले सेयर गरिरहेछ क्या हो सर so that
अ, त्यस्तो कसैले सेयर गरेको त छैन सर, मैले केही सेटिङ
okay
गर्नुपर्छ
okay
कि?
okay okay sir okay भयो sir like I got it.
ओके ओके, यस, वी क्यान सी द स्क्रिन नाउ।
Okay, so let me do it down. So I think like in Nepali or English, like it's fine because all of us are from Nepal, I hope. So none of the participants are from foreign country, I hope. So that I can go with Nepali language also. It will
हुन्छ
be
हुन्छ
better to
शङ्कर सर।
explain, I think. So yeah, so please let me know that one is visible
या इट्स
and
गुड।
slide is visible. Slide so more. Okay.
या इट्स गुड।
So सबैलाई visible छ है I think. Yeah. Okay, so quantitative structure activity relationships in computer-aided drug design. So let's go through what is QSR. So म चाहिँ के assume गरिरहेको छु भन्दाखेरि चाहिँ like all of the participants are from the at least science background or the pharmacy background, so they are quite familiar with the term QSAR. Okay, so and then they know the basic things. So I assume that one. So that's why like I'm not going to take more time in this one. So like as we all know, the QSAR means like it's quantitative structure activity relationship. So it's basically the mathematical equations to describe the biological activity in relation with the uh, physiochemical properties or the or the like descriptors or the molecular descriptors. So how the physiochemical properties or the fingerprint descriptors or the molecular descriptors contributes to the biological activity of that compound. So that is the QSR. So it's basically the mathematical solve of the biology. So हामीले जति पनि हेऱ्यो भने के हुन्छ भन्दाखेरि चाहिँ like biology is quite complicated, so it's very hard to solve, right? So that's why like जहाँ चाहिँ बायोलोजीमा चाहिँ म्याथ पनि त्यति धेरै एक्सप्लेन गर्न सक्दैन, सो त्यो कुरालाई चाहिँ इजी वेमा चाहिँ रिप्रेजेन्ट गर्नको लागि चाहिँ द कन्सेप्ट अफ क्युएसआर इन्ट्रोड्युस भएको हो, जसले चाहिँ के गर्छ भन्दाखेरि चाहिँ हाम्रो ड्रगले देखाउने अथवा कुनै पनि कम्पाउन्डले देखाउने एनी टाइप्स अफ एक्टिभिटिज। सो हामी टुडे चाहिँ हामीले चाहिँ लाइक एक्टिभिटिज बेस्ड मात्रै गरिरहेछौँ यो क्युएसआर, बट एट द सेम टाइम क्युएसआर अल्सो एक्सप्लेन अबाउट द टक्सिकोलोजिकल प्रोपर्टिज। ओके, so like we can also find out the structural relations to the toxicity. Okay, so which part is more contributing in the toxicity of that particular can also be derived from the QSR equation. So but our main focus of this webinar is not about the toxicity but about the biological activity which help to cure the disease or give the a response to the body. So that's why so there is a simple mathematical equations to understand the QSR. So PA means like let's say the biological activity, so it is the function of descriptors. Okay, so descriptor भन्नाले चाहिँ के बुझिदिनु पर्यो भन्दाखेरि चाहिँ so it's a chemical properties at the same times like fingerprint properties and at the same times like it's a quantum characteristics of the compound. So तीनटा कुरा चाहिँ बुझ्यो भने चाहिँ बुझ्न सजिलो हुन्छ डिस्क्रिप्टर भनेको चाहिँ it is not always about the physiochemical properties, sometimes it's about the quantum chemical properties also. So like three-dimensional structures or how the electronic configurations contributes to the biological activity. So in broad sense, if we want to understand, like we have to consider all those parameters. So I think हाम्रो undergrad level मा bachelor level मा चाहिँ we more study about the physiochemical properties, and we try to find out the relations of physiochemical properties in response to the biological activity. But in broader sense, physical physiochemical properties is only the 5% of overall characteristics of that compound. So they were saying QSAR मा चाहिँ quantum characteristics, electronic characteristics, and fingerprint characteristics also play a very good roles. So QSR को हिस्ट्रीमा चाहिँ Hans scientist हुनुहुन्छ, like he is considered as a father of like modern QSR technology, जसले चाहिँ around 60s को टाइममा चाहिँ he explained the lipophilicity, electronic properties, and steric effect to relate the activity. So he derived a equation. So this equations is generally for the this is for linear regression, for the linear regression data. So he derived this one. So log 1 by C. So log 1 by C is considered as a biological activity. So this is the constants, and then this is log p value, so lipophilicity, and then this is sigma. This is draft equations, or you can say like K3 and ES, like it's a steric effect plus some constant. So if you add all these parameter, so that will contributes to the biological activity. So that's what it means. Okay, so log p value also contributes, and sigma also contributes. So it's draft equations, we can say. So this is the steric effect also contributes to the biological activity. So he derived these equations around in 60s. I think that's all about the basic things about the QSR. Okay, so in diagrammatic way, how we can understand it. So suppose we have thousands of compounds or hundreds of compounds. Okay, so uh, and then those compounds activities uh, are known. So known activities छ त्यो कम्पाउन्डहरूको हैन, अनि त्यो नन एक्टिभिटीलाई युज गरेर हामीले QSR को मोडल डेभलप गर्छौँ है. सो इट्स अ मोडल डेभलपमेन्ट प्रोसेस. अनि त्यो मोडल डेभलप गरिसकेपछि हामीले एउटा इक्वेसन पाउँछौँ, जुन मैले प्रिभियसली एउटा इक्वेसन देखाएको थिएँ. सो द्याट टाइप अफ इक्वेसन्स वी क्यान गेट फ्रम द मोडल डेभलपमेन्ट्स. एन्ड नाउ द्याट सेम इक्वेसन्स वी विल युज टु फाइन्ड आउट द एक्टिभिटी अफ अननोन कम्पाउन्ड्स. ओके, सो हामीसँग एउटा अर्को कम्पाउन्ड डिजाइन गऱ्यौँ. अब हामी चाहिँ प्रिडिक्ट गर्न सक्छौँ कि त्यो कम्पाउन्डले कस्तो एक्टिभिटी देखाउन सक्छ त. So this is the overall purpose of doing QSR development. Okay, so the basic this on the head say what we have to understand is first we should have the compound whose activity is already known. त्यो भन्न खोजेको मतलब 100 वटा कम्पाउन्ड छ, त्यसको IC50 भ्यालु चाहिँ हामीलाई थाहा हुनु पर्यो क्या. मतलब अथवा चाहिँ त्यो थाहा हुनु पर्यो इन अ सेन्स लाइक आइदर वी क्यान टेक दोज एक्टिभिटिज फ्रम अनलाइन डेटाबेस अर पब्लिस्ड आर्टिकल्स. होइन, पब्लिस्ड आर्टिकल्स हुनसक्छ, त्यो आर्टिकल्समा IC50 भ्यालु रिपोर्ट गरेको हुनसक्छ होइन. सो त्यो कम्पाउन्ड लग्ने होइन, अनि त्यसपछिलाई चाहिँ हामीले QSR को टेक्निक युज गरेर मोडल डेभलप गर्ने. And then on the basis of that models, now you can design a new compound. Okay, so new compound, and then now you use the same models, then find out the activity or predict the activity of those newly designed compound. Okay, so that like it is going to helpful to us, like which compound may probably show the good activity, so that we can go with that compounds and do the synthesis and again validate by performing the experimental research. So त्यो चाहिँ एउटा process हो है. So in a sort. Okay, so the brief history is there. I will share the slide to everyone so that you can go through it. I don't think like at this stage it is important to study all this history. So yeah, of course, so it started from early 18th, 19th century. So 1863, and then like actually the main things developed in 1962 by Crown Haynes, and then afterwards like a lot of free and Wilson's models came because some of the drawback was there in Haynes equations also, and then he uh, Free and Wilson's derived another equation. So where they propose the additive properties. So additive properties भन्नाले चाहिँ जति पनि डिस्क्रिप्टरहरू छ त्यो सबै डिस्क्रिप्टरले चाहिँ बायोलोजिकल एक्टिभिटीमा केही न केही रोल प्ले गरेको हुन्छ भन्ने इक्वेसन डिराइभ गर्नुभएको थियो एन्ड देन आफ्टर द्याट लाइक स्लोली 80s अर्ली 80s एन्ड अर्ली 90s सम्म आइपुग्दाखेरि चाहिँ हामीले 3D QSAR को कन्सेप्टसम्म पुगिसकेका थियौँ जहाँ चाहिँ कम्फा एन्ड कम्सा लाइक कम्पेरिटिभ मोलिकुलर फिल्ड एनालाइसिस भन्छ जुन चाहिँ 3D QSAR इक्वेसन डेराइभ गर्नको लागि चाहिँ युज हुन्छ मोस्टली चाहिँ अनि अर्को एउटा अप्रोच हुन्छ कम्सा भन्ने हुन्छ हैन सो द्याट इज अल्सो 3D QSAR मोडल अप्रोच गर्नको लागि चाहिँ सो 90s सम्ममा आइपुग्दाखेरि यो कुरा डेभलप भइसकेको थियो एन्ड देन 2000 मा आइपुग्दाखेरि बुझ्नुपर्ने कुरा एकदमै धेरै के हो थियो भन्दाखेरि चाहिँ टिल देन देयर वाज नो रेगुलेटरी बडिज ओके सो because like the process was computational so it is not experimental or the lab ma hune kunai pani in vitro or in vivo experiment just थिएन because all the things used to be done in computer based things or the calculation based things theoretical based things so that like regulations को part हरु थिएन सो अर्ली 2000s मा आइपुग्दाखेरि चाहिँ एकदमै ग्रो हुन थाल्यो QSAR को कन्सेप्ट सो द्याट लाइक दे प्रपोज अ गाइडलाइन विच इज नोन एज OECD guidelines है ना so like economic corporations development guidelines organizations up so that guidelines derive भयो so now all the QSAR models have to follow these OECD guidelines till now हामीले अहिलेसम्म पनि यही गाइडलाइन ल्याउनुपर्छ अनि 2007 मा एकचोटि फेरि यो चेन्ज भएको थियो OECD गाइडलाइन त्यसमा नयाँ कुराहरू थपेको थियो एप्लिकेबिलिटी डोमेनको कुराहरू इन्क्लुड गरेको थियो हैन so now we are following 2007 updated OECD guidelines to develop a QSAR model so त्यो चाहिँ रेगुलेटरी बाटोमा आएको थियो 2000 मा एन्ड देन 2000 देखि 2010 सम्ममा चाहिँ like during that big data era aako थियो that times like the the server and then like data collections data storage ko concepts haru aisakeka थियो and then after that after 2010s like AI and machine learning ko kuraले आइराख्दाखेरि चाहिँ so अहिले so most of the QSAR software haru पनि आफुलाई चाहिँ AI र ML based मा चाहिँ डेभलप गर्दै गइरहेछ Yeah, so this is the basic things like how we can do the or how AI-based QSAR transform the drug discovery process. So initially stage hit and trial and then classical QSAR just linear modeling, simple descriptor we use bio, and then after that they added the AI in it, and then after that the advanced statisticals or algorithms have been used like ML, TL, deep learnings, machine learning, and graph models also they added. And then now they incorporated with the big data and then now like personalized drug designs also can be done on the basis of QSAR study by using the ML and AI. So yeah, so that was the basic the history regarding the QSAR. So now moving forwards, so what are the components or what actually we need to derive models? Okay, so these three things are very, very important. Okay, so especially for the undergrad students or the participant today, so they can take a note on it. So like what we need actually to develop a QSAR models is first things we need the molecular descriptors. So molecular descriptors, so basically they are the numericals values. Okay, so the numericals values just let's say chemical properties and then like physical properties and then quantum properties like say numericals value ma represent गर्छ जसलाई चाहिँ हामी molecular descriptor भन्छ. Okay, let's say the lipophilicity log p भयो हैन कुनै पनि compound को lipophilicity minus let's say like 2.0 छ भने त्यो 2.0 is a molecular descriptors for that lipophilicity. So त्यसरी चाहिँ बुझ्नु पर्यो. So at the same times like quantum properties पनि हुन सक्छ like HOMO, okay, highest occupied molecular orbitals, or LUMO, okay, lowest unoccupied molecular orbitals. So these things also play a big roles in biological activity of that particular compound. So त्यसलाई पनि हामीले the numericals value मा represent गर्यौं भने त्यसलाई चाहिँ we can say the molecular descriptor. So त्यो चाहिँ चाहियो हैन. सो कहाँबाट पाउँछौं त मोलिकुलर डिस्क्रिप्टर भन्दाखेरि चाहिँ हाम्रो डेटा सेटबाट हाम्रो नन डेटा सेटबाट हामीलाई चाहिँ जसको एक्टिभिटी थाहा छ सो त्यो डेटा सेटबाट चाहिँ यो भ्यालु हामीले पाउँछौं. ओके, सो अनदर थिङ्स इज रेस्पोन्स भेरिएबल. सो रेस्पोन्स भेरिएबल्स इट्स एक्सपेरिमेन्टल डेटा जसलाई चाहिँ बायोलोजिकल एक्टिभिटी भनिरहेको छु म अघिदेखि नै. सो त्यसलाई चाहिँ हामी रेस्पोन्स भेरिएबल भन्छौँ हैन, जसलाई चाहिँ वाइ एक्सिसमा राख्न सकियो, जुन चाहिँ एक्सपेरिमेन्टल डेटा हो, जुन भ्यालु चाहिँ एक्सपेरिमेन्टबाट आएको हुनु पर्यो क्या हाम्रो, जसमा चाहिँ हामीले के लिन सक्छौँ भन्दाखेरि चाहिँ बाइन्डिङ एफिनिटी KI or the IC50 value, but in a form of like PIC50 value, we can say. Or if you are designing the toxicity, then you can take a toxicity marker. So that is the response variable. So that means it's the actual data or the actual experimental value which we are going to take either from the database or from the published articles. Okay, so कुनै एउटा साइन्टिस्टले अलरेडी त्यो गरिसकेको छ र रियल भ्यालु दिएको छ, सो त्यो भ्यालु हामीले लान्छौँ. And then third and the most important part of QSR is algorithms. Okay, so the algorithm भन्नाले मानव हामीसँग कम्पाउन्ड पनि छ, त्यो कम्पाउन्डको एक्टिभिटी पनि छ हैन. सो मोलिकुलर डिस्क्रिप्टर छ त्यो कम्पाउन्डबाट चाहिँ. सो मोलिकुलर डिस्क्रिप्टर त हाम्रो 10 वटा 15 वटा हुँदैन क्या. मोलिकुलर डिस्क्रिप्टर्स भनेको चाहिँ मिनिमम पनि 2000 वटा मोलिकुलर डिस्क्रिप्टर लान्छ हामीले. सो लाइक अहिलेसम्म हामीलाई थाहा भएको फिजियोकेमिकल प्रोपर्टिजहरू त्यो गरेर यताउता 10-20 वटा होइन डिस्क्रिप्टरहरू थाहा होला. तर क्युएसआर डेभलप गर्नको लागि चाहिँ हामीले मिनिमम पनि लाइक 1500 देखि 2000 वटा मोलिकुलर डिस्क्रिप्टर लान्छौँ क्या. अब त्यो 2000 वटा मोलिकुलर डिस्क्रिप्टरमा कुन चाहिँ मोलिकुलर डिस्क्रिप्टरले चाहिँ यो एक्सपेरिमेन्टल बायोलोजिकल एक्टिभिटीलाई हेल्प गरिरहेछ त भन्ने कुरा त त्यसै हेरेर त थाहा हुँदैन नि त हैन. सो नर्मल प्रोसेसबाट थाहा हुन सक्दैन. सो फर द्याट रिजन्स वी रिक्वायर the different algorithms. So those algorithms are based on mathematical equation or statistical approach. हैन. सो दुई किसिमले बेस्ड भएको छ त्यो अल्गोरिथम जसले चाहिँ हामीलाई यो कम्प्लेक्सिटी बिट्विन यो दुईटाको रिलेसनशिप्सलाई चाहिँ सल्भ गर्न हेल्प गर्छ. सो एउटा हामीले के लिन सक्छौँ, स्ट्याटिस्टिकल मेथड लिन सक्छौँ. अर्को चाहिँ अहिलेको न्यू ट्रेन्ड्समा चाहिँ अथवा चाहिँ न्यू एडभान्स डिजाइनिङमा चाहिँ मेसिन लर्निङका टेक्निक्सहरू लिन सक्छौँ. लाइक multiple linear regression, so MLR. So multiple linear regression is a statistical approach. Okay, so if you are using the MLR, so that is a statistical approach. So हामीले युज गर्ने सफ्टवेयरमा पनि हामी MLR युज गर्छौँ हैन. सो भोलि जुन सफ्टवेयर युज गर्छौँ. अनि अनदर वान इज सपोर्ट भेक्टर मेसिन. सो यो चाहिँ फेरि मेसिन लर्निङको एल्गोरिथम छ हैन. सो आजको सफ्टवेयरमा चाहिँ हामीले यो मेसिन भेक्टर सपोर्ट भेक्टर मेसिन्स लिन युज गर्छौँ र र्यान्डम फरेस्ट पनि युज गर्न सक्छौँ. सो दिस टु approach represents the machine learning techniques to solve these problems, यो दुईटा बिचको प्रब्लम हैन. And then multiple linear regressions or simple linear regressions or non-linear regressions, multiple non-linear regressions. So यो सबै मेथड भनेको स्ट्याटिस्टिकल मेथड हो जसले चाहिँ यो दुईटा बिचको रिलेसन फाइन्ड आउट गर्न हेल्प गर्छ हैन. सो यो चाहिँ मेन कुरा हो हामीले थाहा पाउनु पर्ने. या आई थिंक इट्स more than enough. So then what is the workflow? And so I think you can take a screenshot for this one. So हामीले डिजाइन गर्दाखेरि चाहिँ यही कुराहरुलाई चाहिँ गर्ने हो है. So what we need भन्दाखेरि चाहिँ first we need the data collections. Okay. सो डेटा कलेक्सन्स भनेको चाहिँ अघि नै जुन फर्स्टमा भनेको थिएँ कम्पाउन्ड थाहा छ जसको चाहिँ एक्टिभिटी पनि थाहा छ, स्ट्रक्चर पनि थाहा छ, लाइक स्माइल फर्मेटमा लिनु पर्यो हैन. अनि जसको एक्सपेरिमेन्टल एक्टिभिटी पनि थाहा हुनु पर्यो, जुन चाहिँ कहाँबाट लाने भन्दाखेरि चाहिँ आइदर फ्रम द रिपोजिटरीज हैन, पब्लिश डेटाबेस, पब्लिकली अवेलेबल डेटाबेसबाट, जस्तै पबकेमहरु भयो हैन, ड्रग ब्याङ्क भयो अथवा केम्बल भयो, त्यस्तोबाट लाने. अर्को भनेको चाहिँ ल त्यस्तोबाट लान अलि गाह्रो भयो भने तपाईहरुले चाहिँ लाइक कुनै पनि पब्लिश लिटरेचर हेर्न सक्नुहुन्छ केमिस्ट्री रिलेटेड जहाँ चाहिँ एक्टिभिटी लग गरेको छ. अनि त्यो हेर्ने त्यहाँ कतिवटा कम्पाउन्डको एक्टिभिटी हेरेको छ, मानव 20 वटा 30 वटा भने सो 30 वटा कम्पाउन्ड लाने, त्यसको एक्टिभिटी के छ त्यो पनि रिपोर्ट गर्ने, स्ट्रक्चर ड्रा गर्ने. सो त्यो प्रोसेसलाई चाहिँ हामी डाटा कलेक्सन भन्छौँ है. सो अनदर वान इज द डिस्क्रिप्टर क्याल्कुलेसन. सो यो रिपोजिटरीबाट त लग्यौं, त्यसपछि सेकेन्ड प्रोसेसमा के गर्नुपर्छ त त्यो कम्पाउन्डहरुको चाहिँ either through smile formula or by using any drawing software. You have to use we have to draw the 3D structures of that particular compounds. And very important steps भनेको त्यो स्ट्रक्चर मात्रै ड्रा गरेर भएन, त्यो स्ट्रक्चर चाहिँ 3D मा ड्रा गर्नुपर्छ. and more importantly we have to minimize the energy of that particular structure. So there are different types of force field available to minimize the energy. So basically we are using like MMF 94 force field, or like some of the research article use DFT. So actually DFT use garyo bhane chai which is better, but it's like so much of resource consuming. So normal laptop ma usma garna chai अलि गाह्रो हुन्छ. So DFT भनेको डेन्सिटी फंक्शनल थ्योरी भन्छ हैन. सो लाइक जसले चाहिँ क्वान्टम मेकानिक्स फलो गर्छ, क्वान्टम क्यारेक्टरिस्टिक्सहरू फलो गर्छ। अनि त्यो गरिसकेपछि त्यसपछि बल्ल हामीले चाहिँ डिस्क्रिप्टरहरू क्यालकुलेट गर्ने। डिस्क्रिप्टर भनेको चाहिँ भ्यालु क्यालकुलेट गर्ने त्यो होइन हाम्रो क्यारेक्टरिस्टिक्सहरूको चाहिँ 2D डिस्क्रिप्टर पनि हुनसक्छ, 3D मोलिकुलर डिस्क्रिप्टर पनि हुनसक्छ। यति भइसकेपछि हामीसँग डेटा पनि भयो, डिस्क्रिप्टर पनि भयो, इनर्जी मिनिमाइज पनि गऱ्यौँ होइन। अब बल्ल चाहिँ हामीले मोडल डेभलप गर्ने होइन। मोडल डेभलप गर्नुभन्दा पहिला चाहिँ एउटा कुरा थाहा हुनुपर्छ, हामीले त्यो डेटालाई चाहिँ हामीले दुईवटा सेटमा डिभाइड गर्नुपर्छ क्या। डेटा भन्न खोजेको मानौ 50वटा कम्पाउन्ड लग्यौँ हामीले होइन। सो त्यो डेटा भयो क्या है, त्यो 50वटा कम्पाउन्ड लग्यौँ। अब त्यो 50वटा कम्पाउन्डलाई चाहिँ हामीले दुईवटा सेटमा डिभाइड गर्नु पर्यो। एउटा सेटलाई ट्रेनिङ सेट भन्छ, अर्कोलाई टेस्ट सेट भन्छ होइन। सो यसको रेसियो चाहिँ जेनरली 70:30 हुन्छ, अथवा चाहिँ 75:25 रेन्जमा हुनुपर्छ भनेर चाहिँ 50वटा मानव 100वटा कम्पाउन्ड लानुभयो भने 70वटालाई चाहिँ तपाईँले केमा मान्ने त ट्रेनिङ सेट मान्ने, बाँकी रहेको 30लाई चाहिँ टेस्ट सेट मान्ने होइन। अब यसलाई कसरी डिभाइड गर्ने भन्दाखेरि चाहिँ अब देयर आर डिफरेन्ट अप्रोच टु डिभाइड इट। ओके, सो आइदर दिस अप्रोच यु क्यान युज and then divide randomly, so which is known as like random approach, so that is also fine. Otherwise like some other machine learning approach are there हैन। सो त्यसको बेसिसमा पनि हामीले यो सेट डिभाइड गर्न सक्छौँ। त्यो डिभाइड गरिसकेपछि चाहिँ मोडल डेभलपमेन्ट गरिन्छ। अनि चाहिँ यो हामीले किन डिभाइड गरेको भन्दाखेरि चाहिँ हामीले मोडल डेभलप गरेर मात्रै भएन, त्यो मोडललाई चाहिँ हामीले भ्यालिडेट पनि गर्नुपर्छ क्या होइन। अदरवाइज लाइक द ओभरफिटिङ हुने चान्सेस चाहिँ एकदमै धेरै हुन्छ QSAR मोडल डेभलप गर्दाखेरि चाहिँ। सो त्यो ओभरफिटिङ नहोस् भनेर चाहिँ भ्यालिडेसन गर्नुपर्छ। सो फर द्याट टेस्ट सेट चाहिन्छ क्या हामीलाई, सो भ्यालिडेसनको लागि चाहिँ। सो मोडल भ्यालिडेसन गर्दाखेरि चाहिँ दुईवटा कुरा ध्यान दिन्छ हामीले चाहिँ। वान इज इन्टरनल क्रस भ्यालिडेसन, सो यो चाहिँ टेस्ट सेटबाट गर्छ। अनि अर्को भनेको एक्सटर्नल भ्यालिडेसन्स, यो भनेको चाहिँ हामीले चाहिँ अरू सेट लाने, अरू कम्पाउन्डहरू लाने, अनि त्यसको एक्टिभिटी चाहिँ चेक गर्ने, प्रिडिक्टेड एक्टिभिटी चाहिँ एक्सपेरिमेन्टको नियर छ कि छैन भन्ने कुरा। अनि एट द एन्ड चाहिँ अब मोडल डेभलप गऱ्यौँ, भ्यालिडेट पनि गऱ्यौँ, सबै क्राइटेरिया लाइक ओआरसिडी गाइडलाइन जुन थियो नि, त्यो ओआरसिडी गाइडलाइन फलो गरेको छ, कट अफ भ्याल्यु एकदमै विदिन द रेन्ज छ भने चाहिँ हामीले ल हाम्रो मोडल चाहिँ भ्यालिडेट भयो। अब यो मोडललाई चाहिँ युज गर्न सक्छौँ र हामीले जति पनि नयाँ कम्पाउन्ड डिजाइन गऱ्यौँ त्यो कम्पाउन्डको IC50 भ्यालु चाहिँ प्रिडिक्ट गर्न सक्छौँ भने 5th स्टेपमा चाहिँ त्यो प्रिडिक्सन गर्ने काम चाहिँ हामीले गर्छौँ द सेम इक्वेसन युज गरेर। सो दिस इज अल अबाउट द QSAR प्रोसिजर है। सो यति कुरा नै फलो गर्ने हो। या आई थिङ्क द्याट्स इट। सो द सेम थिङ्स लाइक आई हेभ गिभन इन यु नो इक्जाम्पल लगेर दिएको छ। ओके, सो ट्रेनिङ सेट टेस्ट सेट डिभाइड गरेको छ होइन, मेसिन लर्निङ डिफ्रेन्ट अल्गोरिदम युज गरेको छ, एन्ड देन त्यहाँबाट आएको IC50 भ्यालु लगेको छ। अनि त्यसलाई चाहिँ लगेर फेरि न्यू कम्पाउन्ड डिजाइन गर्यो यहाँ होइन। सो त्यो न्यू कम्पाउन्डलाई चाहिँ फेरि जुन यो डेभलप गरेको मोडल थियो त्यो मोडलको इक्वेसनमा राख्ने यो न्यू कम्पाउन्डलाई चाहिँ, अनि त्यसपछि IC50 भ्यालु प्रिडिक्ट गर्ने। या सो द्याट्स इट। Here I have written stage wise. Okay, so this is the same things you can know. So एउटा कुरा मात्रै भन्छु यहाँ, क्युरेटेड केमिकल डेटा सेट भनेको छ जुन चाहिँ हामीले सुरुमा भन्यौँ नि, त्यो डेटा सेटहरू हामीले क्लिन गर्नुपर्छ क्या। हामीले जति पनि डाउनलोड गर्छौँ अनलाइनबाट चाहिँ त्यो डेटा सेटहरू धेरै कुराहरू त्यहाँबाट हटाउनु पर्ने हुन्छ क्या होइन। सो त्यो क्युरेसन कसरी गर्ने, क्युरेट कसरी गर्ने भन्ने कुराहरू चाहिँ यहाँ दिएको छु। I request like you can take a screenshot and then go through it so that it will help and हामीले डेभलप गर्दाखेरि चाहिँ। Yeah, this is also the same things जुन मैले अघि एक्सप्लेन गरिसकेँ। ट्रेनिङ सेट टेस्ट सेट्समा डिभाइड गर्ने, जुन चाहिँ टेस्ट सेट चाहिँ हामीले चाहिँ एक्सटर्नल भ्यालिडेसनको लागि चाहिँ युज गर्छौँ भन्ने कुराहरू हो। या द सेम थिङ्स बट इन डिटेल वेमा दिएको छ यसमा चाहिँ। यो पनि म स्लाइड सेयर गर्छु, तपाईँले हेर्नु होला। ओके, सो अघि नै माथि पनि केही अप्रोचहरू दिएको थियौँ। एक्चुअली मोडल डेभलप चाहिँ कुन कुन अप्रोचबाट गर्न सकिन्छ भन्दाखेरि चाहिँ एल्गोरिदमहरू चाहिने। अघि नै भन्यौँ MLR भइहाल्यो। MLR कसले युज गर्छ, QSAR भन्ने चाहिँ सफ्टवेयर हो है, हामीले भोलि युज गर्छौँ यो। र प्लस एक्सेलबाट पनि तपाईँले QSAR डेभलप गर्न सक्नुहुन्छ है। एक्सेलमा पनि मल्टिपल लिनियर रिग्रेसनको अप्रोच लगाउन सक्नुहुन्छ। यु क्यान डेभलप द मोडल थ्रु एक्सेल, बट लाइक भ्यालिडेसन गर्न चाहिँ गाह्रो हुन्छ। त्यही भएर एक्सेलको रिजल्ट चाहिँ त्यति धेरै रिलायबल चाहिँ हुँदैन। एक्सेलमा चाहिँ एउटा एड गर्नुभयो भने पनि गर्न सकिन्छ। एउटा एडिङ्स त्यो गऱ्यो भने मैले सुरुमा सिक्दाखेरि त्यसरी नै गरेको थिएँ। त्यो एउटा भयो। अर्को अप्रोच भनेको पार्सियल लिस्ट स्क्वायर भन्छ, पिएलएस। सो यो चाहिँ केम मास्टर भन्ने एउटा सफ्टवेयर छ, एकाडेमिक युजको लागि फ्री छ होइन। सो केम मास्टरले चाहिँ पार्सियल लिस्ट स्क्वायर एल्गोरिदम युज गर्छ टु डेभलप द मोडल होइन। अर्को प्रिन्सिपल कम्पोनेन्ट रिग्रेसन। सो मैले अहिलेसम्म यो प्रिन्सिपल कम्पोनेन्ट रिग्रेसन अप्रोच युज गर्ने सफ्टवेयर चाहिँ युज गरेको छैन। सो बट दिस इज अल्सो अ भेरी गुड अप्रोच। अनि अनदर वान इज ऱ्यान्डम फरेस्ट, जुन आरएनए क्युएसएआर छ यो सफ्टवेयरको नाम हो, जुन हामीले आजै युज गर्छौँ। सो यो सफ्टवेयरले चाहिँ ऱ्यान्डम फरेस्ट अप्रोच युज गर्छ। अनि द आरएनए क्युएसएआर सेम सफ्टवेयरले चाहिँ सपोर्ट भेक्टर मेसिन पनि युज गर्छ। र अर्को एकदम इम्पोर्टेन्ट भनेको आर्टिफिसियल न्युरल नेटवर्क भन्छ, एएनएन। सो यो एएनएन इज अनदर मेसिन लर्निङ एल्गोरिदम जसले चाहिँ क्युएसआर मोडल डेभलप गर्न सक्छ होइन। So this is also easy one, but like we need the MATLAB. So I think तपाईँहरू सुन्नुभयो होला MATLAB भन्ने सफ्टवेयर। सो this is not freely available, so but around 15-20 हजार जस्तो कस्ट पर्छ पर इयर को सब्सक्रिप्सनको लागि चाहिँ। सो एनिथिङ यु क्यान डु इन MATLAB। सो थ्रु कोडिङ टु स्ट्याटिस्टिकल एनालाइसिस टु एनएमआर को डेटा के भन्छ स्पेक्ट्रा असाइनमेन्ट गर्नुदेखि लिएर मास स्पेक्ट्रोस्कोपीको डेटा असाइनमेन्ट गर्नदेखि लिएर एनिथिङ यु क्यान डु इन दिस MATLAB। सो एन्ड देन आर्टिफिसियल न्युरल नेटवर्क पनि तपाईँले गर्न सक्नुहुन्छ होइन। के नियरेस्ट नेबर, सो हामीले क्युएसआरएन भन्ने सफ्टवेयरमै गर्छौँ यो पनि। डिसिजन ट्री इज अल्सो एभाइलेबल इन क्युएसआरएन सफ्टवेयर, दिस आरएनए क्युएसआर सफ्टवेयरमा एभाइलेबल छ। ऱ्यान्डम फरेस्ट आई अलरेडी टोल्ड, सो आरएनए क्युएसआरमा एभाइलेबल छ। डिप न्युरल नेटवर्क, सो वी क्यान डु इन MATLAB। सो दिज आर द बेसिक थिङ्स। ओके, सो अब एउटा कुरा चाहिँ के कन्फ्युजन हुन्छ होला भन्दाखेरि चाहिँ यति धेरै अप्रोचहरू छ होइन, यति धेरै अल्गोरिदम्सहरू चाहिँ छ, बट लाइक वाट इज द थिङ्स ओके this one, like which machine learning algorithm or a statistical approach we have to use होइन, कुन युज गर्ने त। सो सबैको युज केसहरू फरक छ है। सो सपोज यु हेभ अ स्मल डेटा छ भने चाहिँ मल्टिपल लिनियर रिग्रेसन युज गर्न सकिन्छ। मानौँ डेटा सेट अफ 100 डेटा सेट अफ म्याक्सिमम 200 छ भने मल्टिपल लिनियर रिग्रेसन्सले चाहिँ बेस्ट रिजल्ट दिन्छ अथवा राम्रो रिजल्ट दिन्छ, रिलायबल हुन्छ होइन। सो पार्सियल लिस्ट स्क्वायर अलिकता मिडियम साइजको डेटा सेट छ, एराउन्ड लाइक 400 सम्मको छ भने यो युज गर्न सकियो होइन। ऱ्यान्डम फरेस्ट र सपोर्ट भेक्टरहरू छ, यो अरू पनि, यो चाहिँ बिग डेटा सेटमा युज गर्न सकियो क्या है, डिसिजन ट्री होइन। सो दिज एल्गोरिदम्स हामीले चाहिँ डेटा सेट एकदमै धेरै छ, मानौँ थाउजन्ड्स ट्रेनिङ सेटहरू छ हाम्रो चाहिँ भने ऱ्यान्डम फरेस्ट इज बेटर द अप्रोच टु डेराइभ द इक्वेसन्स। सो त्यसरी चाहिँ हामीले युज केसहरू फरक छ सबैको, अनि एप्लिकेसन्स प्रिन्सिपल्सहरू पनि स्लाइटली फरक छ होइन। सो त्यो कुराहरू चाहिँ ओके। जुन अघि मैले OECD गाइडलाइन भनेको थिएँ होइन, अर्गनाइजेसन्स फर इकोनोमिक कोअपरेसन्स एन्ड डेभलपमेन्ट, OECD गाइडलाइन्स भनेको थिएँ होइन। सो OECD गाइडलाइन्स भनेको चाहिँ यसले चाहिँ कट अफ भ्यालु दिएको छ क्या, जुन मोडल डेभलप गर्छ त्यो मोडल चाहिँ भ्यालिडेट भयो कि भएन भन्ने कुरा चाहिँ कसरी थाहा पाउने त भन्दाखेरि चाहिँ त्यसको कट अफ भ्यालु छ है। सो यो तपाईँले स्क्रिनसट गर्नु होला, दिस इज भेरी भेरी इम्पोर्टेन्ट। तपाईँलाई यो भ्यालु छ भने, थाहा छैन भने थाहा नै हुँदैन क्युएसआर तपाईँको भ्यालिड भयो कि भएन भन्ने कुरा चाहिँ। ल आर स्क्वायरको भ्यालु चाहिँ जेनरली चाहिँ 0.7 भन्दा माथि हुनुपर्छ है, 0.6 भन्दा माथि इज नट क्वाइट पब्लिसेबल, 0.7 भन्दा माथि छ भने पब्लिसेबल। सो इट सुड बी नियर टु 1, 0.9 भन्दा माथि छ भने चाहिँ एकदमै राम्रो भयो होइन। सो एडजस्टेड आर स्क्वायर द सेम वे, एन्ड देन क्यु स्क्वायर भन्छ यसलाई, लिभ वन आउट क्रस भ्यालिडेसन, त्यो चाहिँ भ्यालिडेसनको लागि छ, जसको भ्यालु चाहिँ 0.6 भन्दा माथि भन्छ, तर 0.7 भन्दा माथि भयो भने राम्रो मान्छ होइन। सो दिस इज अल्सो 0.6 भन्दा माथि भयो भने राम्रो मान्छ। सो लाइक दिस है। सो यो भ्यालु चाहिँ तपाईँले लिनु होला, यो गर्न थाल्यो भने टाइम जान्छ। ओके, स्क्रिनसट गरेर यु क्यान लुक एट इन्टु इट। सो दिज मेनी भेरिएबल्स, दिज मेनी भ्यालुज वी हेभ टु, सो यो सबै भ्यालुहरू चाहिँ हामीले पाउँछौँ है सफ्टवेयरबाट चाहिँ। अनि त्यसको रेन्जहरू यहाँ दिएको छ कति हुनुपर्छ होइन, त्यसको युज केस पनि यहाँ दिएको छ। तपाईँले हेर्नुभयो होला यो वाइ रेन्डमाइजेसन्स भनेको छ। यो वाइ रेन्डमाइजेसन चाहिँ किन गर्छौँ भन्दाखेरि चाहिँ यो बुझ्नुपर्ने कुरा चाहिँ छ, हामीले जुन मोडल डेभलप गर्छौँ त्यो मोडलको मान हो यो माथिको सबै प्यारामिटर्सहरू छ नि एकदमै राम्रो आयो क्या, एक्सिलेन्ट एक्सिलेन्ट प्यारामिटर्सहरू आयो होइन। तर वाइ रेन्डमाइजेसन्स हेर्दाखेरि चाहिँ यो भ्यालु चाहिँ फुलफिल भएन भने हामीले के बुझ्नु पर्यो भन्दाखेरि चाहिँ हामीले डेभलप गरेको मोडल चाहिँ बाई चान्स डेभलप भएको हो क्या। सो टु फाइन्ड आउट द ऱ्यान्डमाइजेसन्स अर द चान्स कोरिलेसन्स कम गर्नको लागि चाहिँ हामीले ऱ्यान्डमाइजेसन्सको भ्यालु हेर्नै पर्छ होइन। योभन्दा माथिको भ्यालु एक्सलेन्ट भएर केही पनि हुँदैन यदि वाइ ऱ्यान्डमाइजेसन्सको भ्यालु चाहिँ एक्युरेट भएको छैन भने। त्यसको मतलब के हुनसक्छ त भन्दाखेरि चाहिँ हामीले डेभलप गरेको मोडल चान्स बाई चान्स भएको हो त्यो एक्चुली, अथवा चाहिँ ओभर फिटिङको प्रोब्लम आइरहेछ भन्ने कुरा चाहिँ बुझ्नुपर्छ, विच इज भेरी इम्पोर्टेन्ट। सो यो ओआरसिडी गाइडलाइनले दिएको कुराहरू चाहिँ हो है। Another guidelines is get there Goldbrick Trofa external validations criteria यो चाहिँ। सो यो माथिको कुराहरू चाहिँ इन्टरनल भ्यालिडेसन्सको लागि चाहिँ युज हुन्छ म्याक्सिमम होइन। सो एक्सटर्नलको लागि पनि युज हुन्छ, तर एक्चुअल एक्सटर्नल भ्यालिडेसन्स क्राइटेरिया चाहिँ यो हो है। सो आर स्क्वायर टेस्टको कति हुनुपर्छ होइन, क्यु स्क्वायर कति हुनुपर्छ, आर स्क्वायर र यो सबभन्दा इम्पोर्टेन्ट भ्यालु भनेको यो दुईवटा भ्यालु। आई थिङ्क दिस भ्यालु इज डेराइभ बाई सम इन्डियन स्ट्याटिस्टिसियन है। सो यो के र के ड्यासको भ्यालु चाहिँ 0.85 देखि 1.15 को बिचमा आउनुपर्छ होइन। यदि यो भ्यालु यो बिचमा छैन भने हामीले डेभलप गरेको मोडल चाहिँ एक्युरेट हुँदैन भन्ने कुरा चाहिँ हो या। सो दिस भ्यालु आर भेरी इम्पोर्टेन्ट। यस, सो सफ्टवेयर र वेब सर्भरहरू के छ त, जस्तै डेटा कलेक्सन भनिरहेको छौँ अघि नै, कहाँ कहाँदेखि गर्ने त, यो स्क्रिनसट लिनु होला तपाईँलाई काम लाग्ला है। केम्बेल भन्ने वेब सर्भर छ, पपकेमबाट गर्न सकियो। बाइन्डिङ डिबी भन्ने छ, ड्रग ब्याङ्क, जिङ, केम स्पाइडर र अथवा चाहिँ पपकेम बायोएसएबाट पनि हामीले त्यो डेटाहरू लिन सकियो। डेटा भनेको आइसी50 भ्यालु र स्ट्रक्चर अफ द कम्पाउन्ड। त्यति लगिसकेपछि त्यो स्ट्रक्चर प्रिपेयर गर्नु पर्यो, अप्टिमाइज पनि गर्नु पर्यो। स्ट्रक्चर आइदर तपाईँहरूले ड्र गर्न सक्नुहुन्छ बाई युजिङ दिस ड्रइङ सफ्टवेयर। केमरा आई थिङ्क फ्री भर्जन हो छैन क्या र होइन। मार्बिन स्केच चाहिँ फ्री हो, एभोगाडो पनि फ्री हो। सो मार्बिन स्केचले चाहिँ तपाईँले ड्र गर्न सकिन्छ स्ट्रक्चर, एभोगाडोमा चाहिँ तपाईँले चाहिँ ड्र पनि गर्न सक्नुहुन्छ प्लस अप्टिमाइजेसन पनि गर्न सक्नुहुन्छ, इनर्जी मिनिमाइजेसन पनि गर्न सक्नुहुन्छ एभोगाडोमा। ओपन बाइबल इज अल्सो फर इनर्जी मिनिमाइजेसन होइन, जुन फोर्स फिल्डहरू छ। एन्ड देन मोस्ट इम्पोर्टेन्ट इज आरडी किट। ओके, सो आई थिङ्क तपाईँले चाहिँ यदि बायो इन्फर्मेटिक्स अथवा चाहिँ कम्प्युटर ड्रग डिजाइन्स गर्न चाहनुहुन्छ भने चाहिँ यु सुड बी फ्यामिलियर विथ आरडी किट। आरडी किटसँग फ्यामिलियर हुनुभयो भने चाहिँ एकदमै राम्रो हुन्छ यो जानिराख्दा। यु क्यान डु एनिथिङ इन आरडी किट्स। ओके, सो दिस इज अ पाइथन बेस्ड कोडिङ ओपन एक्सेस platform where you can do a lot of things related to the chemistry. Okay, so from finding the structure, drawing the structure, finding the descriptors, energy minimizations, everything you can do. So RDKit is very good. अनि कतिखेर के पनि गर्नुपर्छ भन्दाखेरि चाहिँ एउटा लास्ट विक चाहिँ आई गट ए प्रब्लम्स। ओके, सो मलाई एउटा प्रब्लम आयो। आई डिड एभ्रिथिङ एक्युरेटली होइन, सबै कुरा चाहिँ मैले एकदमै एक्युरेट गरिरहेको थिएँ। स्ट्रक्चर पनि ड्र एकदमै एक्युरेट भएको थियो, 3 डाइमेन्सनल कन्भर्जन्स, 3D स्ट्रक्चर पनि भयो, इनर्जी मिनिमाइजेसन गरेँ होइन, सबै कुराहरू फलो गरेको छु। बट स्टिल यो चाहिँ डकिङ गर्दाखेरि चाहिँ स्टिल समवेयर त्यो ड्रग चाहिँ भिजुअलाइजेसन गर्दाखेरि चाहिँ ब्रेक भएको देखिन्थ्यो क्या। अनि पछि सर्च गरेँ मैले लिटरेचरहरू बुझ्दै जाँदाखेरि चाहिँ के भएको रहेछ भन्दाखेरि चाहिँ कहिलेकाहीँ चाहिँ यो ड्रइङ सफ्टवेयरहरू, इनर्जी मिनिमाइजेसन गर्ने सफ्टवेयरहरूले चाहिँ स्माइल नोटेसनलाई चाहिँ अप्टिमम तरिकाले युज गर्दैन। लाइक यो प्रब्लम चाहिँ अब थाउजन्ड्स अथवा चाहिँ 100 अफ थाउजन्ड्समा एकचोटि हुनसक्छ अनि आउन सक्छ, अथवा चाहिँ बट त्यो प्रब्लम आउन सक्छ। त्यस्तो कन्डिसनमा चाहिँ लाइक 3D स्ट्रक्चरलाई चाहिँ राम्रोसँग रिड गरिदिँदैन क्या कहिलेकाहीँ चाहिँ। सो इन द्याट सेन्स वी हेभ टु युज द आरडी किट होइन। सो आरडी किटले चाहिँ एक्युरेटली विदाउट एनी इरर 3D स्ट्रक्चरलाई चाहिँ मिनिमाइज पनि गर्छ, ड्र पनि गर्छ, प्रोपर्टिज पनि थाहा हुन्छ। अनि त्यसमा हुँदाखेरि चाहिँ मैले एउटा सानो स्क्रिप्ट डेभलप गरेको छु पछि यो आइसकेपछि चाहिँ। सो मैले डेभलप गरेँ अनि चाहिँ लाइक आई विल सेयर द्याट स्क्रिप्ट। सो कुनै दिन लाइक इट क्यान बी युजफुल फर यु। सो एन्ड देन लाइक आई गट good results and then visualization चाहिँ राम्रो गर्न सकेँ सो त्यसो हुँदाखेरि चाहिँ आरडी किटसँग चाहिँ फ्यामिलियर भयो भने राम्रो भयो सो दिस इज द ओपन प्लेटफर्म सो हामीले एमडी सिमुलेसन गराएको थियौँ नि जुन गुगल कोल्याबमा सो गुगल कोल्याबमा हामीले आरडी किटको कामहरू गर्न सक्छौँ Another one is descriptor. So descriptor कहाँबाट क्यालकुलेट गर्ने त मैले स्टार्टिङमा भनेको थिएँ we have to calculate the around 1500 to 2000 descriptors. Okay, so from where we can calculate those descriptors. So the very famously used software to calculate the descriptors is Paddle. So it's freely available, but I don't know nowadays. I'm not finding it on the online. So I don't know what happens. Okay, so मैले अनलाइनमा सर्च गर्ने खोजी पाइरहेको छैन, but it's like as a software version it's not available, but like some of the this ChemDash is there, so under the ChemDash server, so these descriptors are available, so we can use the ChemDash. And then so RNA QSARs itself calculate the descriptors, and ChemDash is another web server, so we are going to use this one tomorrow. So from here we will calculate the descriptors, paddle descriptors also. And ChemMasters, this is software, so this software has a limited number of descriptors, so which can be calculated. Okay, so and most important one is RDKit. So if you know a little bit of Python, so a little bit of if you learn the thing, so you can calculate the descriptor using RDKit also. And PyDescriptors is also there, so this is a Python based descriptor. So all of these things are freely available, so okay, no need to pay a single penny for this. Another one is data प्रोसेसिङ गर्नु पर्यो अब डिस्क्रिप्टलाई चाहिँ भन्नाले चुज गर्नु पर्यो कतिपय कुराहरू चाहिँदैन कतिपय कोरिलेसन्स हुने फ्याक्टरहरू हुन्छ कतिपयको भ्यालु 0 हुन्छ कतिपयको डिस्क्रिप्टको भ्यालु 111 हुन्छ त्यो सबै कुरा हटाउनु पर्छ यदि भ्यालु 0 छ भने त्यो सबै हटाउनु पर्यो 1 छ भने त्यो हटाउनु पर्यो म्यानुअली अल्सो वी क्यान डु but we have a software QSAR and tomorrow we will use this one, so in this one automatically it can be done and RNAs in this one also automatically be done. But म तपाईहरुलाई चाहिँ एक्सेल मा चाहिँ म्यानुअली पनि कसरी गर्ने भन्ने कुरा पनि देखाउँछु सिकाइदिन्छु so that in near future anywhere you can use those techniques. Last मा चाहिँ अब मोडल डेभलप भ्यालिडेसन गर्नको लागि चाहिँ these are the softwares which are freely available. Okay, so ChemMaster is also available, this is also freely available, this is also freely available. So these are the software required for QSAR development. Yeah, so this is one examples from our ongoing research projects. So this what models we can say we developed with collaborations with Dr. Devaraj. So उहाँले गर्नुभएको एक्सपेरिमेन्टल वर्कमा चाहिँ मैले QSAR युज गरेर डेभलप गरेको यो मोडल हो हैन सो भन्नाले यो किसिमको मोडल पाउने हो कि हामीले चाहिँ हैन यो भ्यालु छ प्लस यो भ्यालु छ सो दिस इज द डिस्क्रिप्टर तपाईले कहिले पनि देख्नु भएको छैन होला यस्तो पनि डिस्क्रिप्टर हुन्छ भनेर AATS81 भनेको छ हैन AATS660 सो हामीहरुले अहिलेसम्म हेरेको डिस्क्रिप्टरहरु भनेको त हैन लग पी भ्यालु भन्यौं हैन अथवा चाहिँ लिपोफिलिसिटी अथवा चाहिँ मोलिकुलर वेट अथवा चाहिँ लाइक त्यो किसिमको टाफ्ट इक्वेशन्स अथवा ह्यामेट इक्वेशन्स हैन सो त्यो किसिमको कुराहरु चाहिँ गरेको थिएँ आई थिंक माथि मैले सिग्मा भन्ने त त्यसलाई चाहिँ अघि टाफ्ट भनिरहेको थिएँ इट्स अ ह्यामेट ओके सो मेबी प्लिज त्यो सच्याउनु होला सो ह्यामेट इक्वेशन्स त्यो बेसिक कुराहरु मात्र देखिरहेको थियौं तर यहाँ हेर्नु त त्यो बेसिक कुराहरुले चाहिँ हाम्रो एक्टिभिटीमा केही पनि हेल्प नगरेर छ क्या एज पर आवर मोडल्स हैन सो दिस आर द डिस्क्रिप्टर्स हु कन्ट्रिब्युट्स टु the biological activity. So this is the linear regression curve and a multiple linear regression curve as I said test set training set or so these types of equations we will get. So now the same equations we will use in newly designed compounds and then we can calculate the PIC50 value. So that's what we do. Yeah, so this is the software which we will do today sessions. अब लाइक आई थिंक 10 मिनट 15 मिनटमा सकिहाल्छ होला यस्तो गाह्रो छैन यो त्यही भएर त्यो धेरै टाइम लाग्दैन हैन सो दिस इज द इन्टरफेस अफ दैट आरएनए QSAR एन्ड एमएल सुट भन्ने सफ्टवेयर छ हैन यसमा चाहिँ हामीले ट्रेनिङ सेट अपलोड गर्ने त्यो ट्रेनिङ सेटमा के हुनु पर्यो भन्दाखेरि चाहिँ स्माइल फर्मुला र बायोलोजिकल एक्टिभिटी भएको ट्रेनिङ सेट चाहिँ कस्तोमा हुनु पर्यो एक्सेल फाइलमा भयो भने एक्सेल फाइलमा अपलोड गरिदिने अनि यहाँ अरु कुराहरु चाहिँ छन् त्यो कुराहरुलाई चाहिँ क्लिक गर्यो भने लाइक we can develop a models within 1 minute हैन त्यो मोडल डेभलप गरिसकेपछि त्यसको रिजल्टहरु चाहिँ हामीले ORCD गाइडलाइनसँग म्याच गर्ने either the result obtained from those the equations fulfill the criteria given by the ORCD or not, so that we will calculate if it fulfills all the criteria then we can say the developed equations or the models is validated. Then we can use the same models to predict the biological activity of new compounds which were not included in our data. So that's what we are going to do. Okay. So yeah, I think this is all about the theory part of QSR things. Yeah. So I will be happy if any questions is there regarding the things. Otherwise, we will move towards the practical sessions. Yeah. Thank you.
Someone is raising their hand. Please try to put your question in the chat box as possible. If we could not, then raise hand and be very much specific and very quick. Please, Zahid, please go ahead.
Thank you so much. I'm Zahid Yasin. Currently, I'm in the second semester PhD actually. So I opened the LinkedIn and I saw this webinar, so I just joined. So basically, I wanted to include the QSAR. I have very small questions because I'm quite new to QSAR. And my first question is that QSAR and the AI/ML is same or different?
Yeah. So welcome. Yeah, thank you for being participating today. So yeah, so it's a valid question. So one thing we have to understand. Okay, so QSR is a one technique. So to get a QSR, we are using tools. So among them, one is AI/ML. So that's how you can understand. So AI/MLs is a tools who can give you the models. Okay, like statistical tools like multiple linear regressions is one approach to develop the QSR models. In the same way, you can incorporate either AI or machine learning things. Okay, so which will be able to give you the desired QSR model. So and then you can validate it and then use for your future study.
And also all the softwares that you mentioned and all the algorithms, are they like free, open source?
Yeah, yeah. So that's what like our main uh, approach or the motto of the webinar, that so whatever the things we are using here, so those all the things are freely available, so you can
Okay,
download and then you can use
thank you,
those
thank you.
open licensing.
So,
Yeah.
okay, thank you so much. So my other question is are you open to collaborations? Because my guide, he is very new to the AI/ML and all the QSAR. So are you open for the collaboration to guide the
Yeah, okay.
students?
So yeah, please like reach out to me or the Dr. Dirga. We can talk it about later. Yeah. So if any other questions related to today's webinars are there, we're happy to address. So these things we can discuss later. Yeah, you can reach out to me or Dr. Dirga or any of our participants who are willing to the collaboration. So yeah, you can reach out to us later.
Thank you so much. I was actually looking for the guys who can guide me. Thank you.
Yeah.
Thank you, Zahid. It's nice to hear your question. Rabiji, please go ahead. You are asking something.
सर QSAR रन गर्नको लागि हामीलाई हायर ग्राफिक्स कार्डको ल्यापटप चाहिन्छ कि हामी नर्मल ल्यापटपहरुमा पनि गर्न सक्छौं?
नर्मलमा गर्न सक्नुहुन्छ। लाइक मेरो ल्यापटप मैले गर्ने गर्ने ल्यापटप पनि नर्मल तपाईँहरूको जस्तै i5 नै हो होइन, नेभिडियाको ग्राफिक्स कार्ड छ, i5 ग्राफिक कार्ड केही पनि छैन। नर्मल ल्यापटप चाहिँ जुन युज गर्नुहुन्छ त्यसमा गर्न सक्नुहुन्छ। तर कहिलेकाहीँ चाहिँ डिस्क्रिप्टिभ क्यालकुलेसन गर्दाखेरि चाहिँ होइन, तपाईँ हामीहरूले चाहिँ लाइक प्याडल डिस्क्रिप्टिभ क्यालकुलेसन प्याडल सफ्टवेयर युज गर्छौँ। कहिलेकाहीँ चाहिँ लाइक इफ लाइक योर training sets is more than 50 or more than 60, it may takes around 5 to 6 hours to complete the task. Other than that, there is no problems using any normal laptops to develop the QSR models. Yeah, गर्न सक्नुहुन्छ मजाले होइन। अथवा आरडी किट युज गर्न सक्नुहुन्छ, विच इज अल्सो फ्री। तपाईँले आरडी किट चाहिँ गुगल कोल्याबबाट युज गर्न सक्नुहुन्छ, त्यहाँबाट गर्दाखेरि पनि हुन्छ। अथवा चाहिँ नर्मली आरडी किट डाउनलोड गरेर अथवा चाहिँ अरू चाहिने डिपेन्डेन्सीहरू डाउनलोड गरेर पनि गर्न सक्नुहुन्छ। म चाहिँ त्यति धेरै फेमिलियर छैन, सो आई डोन्ट वान्ट टु really talk about it. So I only know the basic things regarding the RTKit, how to draw the structure, how to minimize the things, how to convert into 3D. So those simple parameters I know. And then yeah, it's free platforms, you can use it.
Okay, thank you, sir.
Our next Ram Sahaji, he is raising hand. Please go ahead. Be very
Yes, sir. Good evening, sir.
specific.
I'm Ramshankar Goswami from Srinath College of Pharmacy. Sir, my question is very specific. I'm just asking that you have shown the various, apps or websites or key parameters of like biostats mainly and the QSR approaches. Are there any type of limitations or criteria in specific works or specific research journals? Like if I take an example, like the Scopus Index Journal or Web of Science Journal, if they set a criteria that you can use only these types of computational apps or this type of computational techniques to follow the our guidelines and follow your research work, then you can publish your paper or your article here or your work here. Is there any type of criteria related to it in QSAR if I work in this QSAR field?
No, actually there are no such types of criteria as of my knowledge. And then because I am intensively working on this field, so as of now I didn't found any of journals who set the particular criteria regarding the software on any of the things. So you can use the reliable open access or the open license software and then do the QSR study and build the models and then validate it, and then you can publish your data in Q1 journal as well. So there is no such limitations. But the provided criteria is, so you have to follow the ORCD guidelines. That is most important. So if you follow the ORCD guidelines, and then any softwares or the anything you can use, irrespective of anything, you can publish in high index peer review journals.
Okay, thank you, sir.
Yeah. Yeah, please go ahead. Zaneed is raising your the hand. Yeah.
So I have a very small question. Is there any requirement of any specific, computer specification should be specific like
No.
RAM or graphics card? And also at any level, is there requirement of any supercomputer?
Yeah, particularly for the our level of research, like our levels means like undergrad research or the graduation research level. So if your sample size is about 100 or 200, so there is no requirement of extra, graphic cards or the supercomputers to derive the QSR models. Obviously, if you want to do the drug discovery process where you want to conduct the MD simulations of, let's say, the thousands nanoseconds or MD simulations of 500 nanoseconds. In that case, yeah, you require specific graphic cards, and then you can't do in laptops, and then you need the desktops having the good graphics card supports, and it takes time. So in that sense, it's required, but like for particularly QSR, it our levels of like hundreds to 200 sample size, like the normal laptops which you are using for your daily works is sufficient to do so. Yeah.
One person, one question at a time, please. If there is no more question, if
Sorry
you
to
have
for
any more
interrupting
question,
in this
please put
section.
that on your chat box. Yeah, Ram Shankarji, do you have more question?
Yes, sir. Just one small question, actually.
Yeah.
Uh, Actually, sir, in the cases we have seen here in the computational docking or in the computational QSR model, specifically the more than 90% drugs, when it is comes into the weight lab scale, they're getting failed during the clinical trial. So, any type of reason there or valid reason that when I am seeing in the QSR models or approaches, they seem okay with that, but when the computational jogging is actually scaling up into the practical sessions, so they are getting failed many times, so we have to improve again and again. So is there any valid reason or questions, any type of answer
Yeah,
this?
so yeah. So I think like that's the problems with the in silico study or the computational. So not the problems, it's the limitations, right? So everything has their own limitations. Actually, I wanted to say at the starting of the session, so that like we have to aware about the limitations what you are using. So for these computational things, the limitations is all the data, all the results are predicted results. they are not confirmatory results. Okay, so whatever the results we are getting, so those results are predicted results, either it's from the dockings or QSAR, so whatever the score you are getting. So while writing the manuscripts or analyzing the data, so you have to be very particular uh, for the selections of word, right? So you can't use the word like confirmatory or revealed or based score or based activity. So those types of word you can't use because it's a prediction. So if you use the word accurately, so like the computational result revealed in that way, or the predictory this one's like score of the docking. So in that way you have to write the result. So yeah, so this is the limitations. Yeah, so as you know, every approach has limitations. Computational results sometimes can't be translated into in vitro, and then in vitro results can't be translated into in vivo, and in vivo results can't be translated into clinical aspect, clinical setting. So yeah, for that ones like plenty of factors are there would play to do so. I think that is another topic to discuss. So sometime later we can discuss on that. Yeah, but you have to be very clear to write the things. So it's a prediction. So it's not a confirmation. Yeah.
So can we, sir, start or just boost up a little bit of wet lab scale techniques after the QSAR approaches or QSAR experiments are going in the middle of the session? I mean, it's just on the middle session, and then we can start the wet lab techniques, so we can at least get an idea about that whether it will be druggable or not.
Yeah, from QSR you can. That's what like I'm saying. Like suppose you have taken the hundred of compounds and developed the models, and now you design 10,000s of compounds and use the models to predict the IC50 value, right? So among the 10,000s models, you will suppose you got 10 models as showing the best IC50 value, 10 compounds. So you will go with those 10 compounds and do the wet lab study. First you synthesize and try to find out the activity by using cell line study or and all the things. So in that way it is helpful, right? So you can't completely translate all the results in your experimental values. So all the thousands compounds is never going to give you the same results which you predict, right? So it will make your work a little easier instead of like synthesizing the thousands of compounds. Now you only have to synthesize, let's say 20 or 50. So in that way it is helpful. Uh, in another approach, what you are saying, like in that way, obviously there is a lot of gap in between from computationals to wet lab things. Yeah, so I think with the advanced technology or the advanced technique in near futures, it can be reduced, but I don't think like completely, this problem can't be solved in near future also. Obviously, there will be a gap.
Yeah, there is another similar question from pharmacist survey small rare, but looks like you already answered this. And as the Shankar sir also explained, it is very common here. We do parallelly everything, even molecular docking, MD simulation, QSAR, and side by side, real compound synthesis, validation, in vitro, in vivo assay, correlated, everything are done simultaneously in a bigger drug discovery lab. While I was back in US working at NIH NCATS, I was in the dedicated drug discovery team. There we have a dedicated bioinformatics team who do the AI, machine learning, QSAR. As our team part, we do the docking and other thing and the compound synthesis. So every week we used to have meet together and discuss what came through the docking, what came through the synthesis, and let's progress. So it's a continuous process. it it not like we do one thing then another thing. Earlier when we screen around 15,000 compound to develop the model, the success rate was quite low, 0.5% something. But after QSAR prediction, we raised that the possibility to the 20%, right? That's, it means we decrease the failure rate, and it's drastically changed. So definitely computer is helping, but it's always a need to validate through the in vitro and other assay, because actually we need to do experiment, but it will rule down the uh, less effective or the ineffective molecule, so that we can focus on the major molecule. We can manage with the low resources, save the time, um, everything, right? So I am very much positive with the AI, machine learning, QSAR, everything these days it's helping. That's why the new and new model are developing, right? Shankar sir told some long back decade history. Earlier it was quite simple, 2D, 3D. Now we incorporate other thing. Yeah, anyway, looks like this discussion is quite engaging. If there is any more question or some suggestion from Shankar sir, then we can take that. Otherwise, we will close the webinar today. Please feel free to ask if you have any
Yeah,
question.
yeah, Durga sir, you rightly, mentioned the thing. So we are not going to replace real experiment in vitro or in vivo experiments through the computational power. Okay, so we are going to just helping the in vitro study and then reducing the time cost of drug discovery by giving the better platform. Okay, through this all the in silico approach, either it's dockings or energy minimizations or the free energy calculations or QSAR, or yeah, there is another very good approach to look the drug discovery through the DFT, so density functional theory. So it's basically based on the quantum mechanics. So that also gives very, very authentic results to help the experimental activity or the experimental research. Yeah, so we can't never, or we never like replace the things with the, that's what I feel. Okay, so at the any stage of the drug discovery, we have to do the real research or the clinical research. So that need to be done. So QSR need to be validated at the end by in vitro study. So yeah, so whatever the results we are getting from QSR model is not final result. That's what we have to keep on in our mind. Yeah, so with this, like I think we discuss plenty of things. Now we can move towards the practical sessions. If yeah, if you have a questions, I think at the end of the sessions also we can discuss regarding this these things. Yeah.
Yeah, let's go.
Okay. So as I mentioned in the slide, like first things we need the data or we need the compounds, right? So for that I told one website, or the not website, it's a database. So from there we can take a data. Yes, so before that I will show you how to download the software, required software. uh, as Dr. Dirgasar has given the link in the text, you can go through that first link, that Zenido is there, so you can open that one. Okay, so let me share my screen again once again. Okay, so this is the Zenido website. Actually, this is the repository site. Anyone can upload their data in this one for the repository. For publication things also, you can use this Zenido repository site to keep your raw data. Okay, so most of the times what we do, we generate the file and then final result and make a manuscript and then publish. But what we forgot is we forgot to recover the data. Okay, so in near future, if anyone is asking, so okay, please share the data from where you got this result. So at that times it may not be in your desktop. So for that reasons, to keep your data safe, you can use this Zenido. So I think these researchers also use the Zenido. I must thank all the authors and then developer of this software. Okay, so I'm thankful for them. So they made it open access to everyone. So yeah, so I will show you. So this is the thing. So here at the end you have an option there, file. Okay, so this file options is there, and then here after that name is there, right? So here download all. So you have to download it from here. So you can do the download and then you can download the software. So after downloading, you will be able to get this file. Okay, so let me show you. I downloaded it long back ago. So this file you will get. So the software name is run QSAR. Okay, so run QSAR. So I think the developer is from India. I think so. So it's a new software. Okay, and then the software is based on machine learning and then AI. So these types of things you will get, and then you just open it. Okay, so inside the window, the folder, you will get this many data. So from there, like there is a one file which is called applications. Okay, ran QSR. You can double click it and then install it. Okay, so after installation, and one more things, before doing before conducting your experiment, you can go through this tutorial. So everything is explained in this tutorials, how to do, what type of data it requires, or what are the steps. So everything is, and what every this algorithm means, how you can use those algorithms, it's given there. Okay, so I strongly recommend you to go through it before conducting your experiment. Yeah, so that's how you can do. You can download the server. It's software. It's freely available. After that, you can get this shortcut. Okay, so you just click it, double click, so that you can get it. Yeah, that's all about the software required to download, or the required for today's session. So other software which requires for tomorrow's sessions, I will explain tomorrow itself. Yeah, so before that, let it open. So let me show you how to extract the data, or how to get the data. So one approach I already told. So you can go any of the published articles. So as Dr. Dirgaraj sir share his research publications in the chat box, I think that is also related to in vitro study and then in silico study. So that type of paper will have, you can get it through the online, and then go through their synthesized compound and their IC50 value. So all the compounds you can take, and IC50 value also you can take. So now you draw those compounds using the drawing softwares, and then minimize the energy, and then calculate the descriptors. So and then use it for QSAR model development. So that is one way from the published paper. So another way from publicly available database, how you can get the IC50 value and compound. So for that one database I am going to show you is Campbell, right? CSAMBL. Okay, so this database is there. This is large database. You can get millions of compounds with their biological activity. So always remember, so biological activity should be in IC50 value. Okay, so like you can get in MIC form also, or in any other, or micromolar per, that one in that form also. So but we need in IC50 or KI, like either binding affinity we need. Okay, so or inhibitory concentrations we need, like IC50 we need. So here you can type the term. Okay, so what type of activity you are planning to do uh, on the research, like let's say you are doing on cancer, Alzheimer's, okay, diabetes, or antibacterials, whatever. So in that things also, which targets you are mainly focusing. So in cancer also, like so many targets are there, right? So on those so many targets, so many assay methods are there, and then so in that way you have to be particular to as per your research. So you can type it here. Okay, so like the enzymes assay, you can type it. So let's say the anyone enzymes, anyone can say. So for better understanding, like cancer, like PIM kinase is there, because I work on this and then I published one article. So in this way you can type it. Okay, like PIM kinase inhibitions or inhibitors. Okay, whatever things you can type it, and then you can download. So after that you will get thousands of compound over here. So before that you have to be uh, sure that you are in correct this one, like correct assay types or the target types. Okay, so you can get it compound. So how many compounds? 651 compounds are there, and the target 1.7K, but the assay 647K assay are related to this particular term. Okay, so you have to be in this assay panels. Okay, so if you scroll down, so you can get it plenty of assay related to that PIM kinase inhibitions. Okay, so one thing you have to be sure that what is the assay type. So while choosing the data, or the, you can say, the compounds or the training set, assay type should be same. Okay, so if your assay type is not same, that is not good. Okay, so another one is organisms. So on which organisms your research is based. Okay, so that is very important. Okay, so these two things you have to be sure. And then another one, sometimes like it will show how many compounds they have done in this particular assay. So that is also considerable things. Okay, so here in that way you can do. Okay, so you can go scroll down, so many things are there, so many. So you can go to the next page. Okay, so in that way, plenty of, you know, uh, resource available in that one. So let me show you, like, let's open these things, right? Inhibitions of PIM1 kinase is there. So this is the bindings, but they didn't mention the organisms. Just for demo purpose, let me so open this. Okay, so here you will get the basic informations. Okay, scroll down, curation summary and activity. So this is very, very important. So activity, it is mentioned, right? So here, like, what activity they reported in IC50 forms and how many compounds they have included in this assay. Only 3 compounds they have included, right? So in that way, so suppose you need the 50 compounds, so you can go for another, okay, headings or another thing, so that you can get, or one by one you can collect. Let's say 3 compounds you are collecting from this assay, the remaining other compounds you can collect from another headings. But the provided conditions is the assay type should be same. Okay, your enzyme should be same, and those all parameters should matched. So let's say open. So after that, what you have to do, you have to just click here. Okay, in this IC50 value, you have to click. Yeah, so once, so it will direct it towards the compound. Okay, so their 2D structure, it will direct. And then along with that, you will get other informations also. Okay, so let's say the here, like, standard relations, standard value. So this value is very important. So what I was talking during the theory part, so I was talking about the biological activity. So this is standard value is your biological activity. So this is IC50. Okay, this is standard value. So this is your biological activity. So this value we have to note down. Okay, and then this value is given by this compound, and this IC50 value is given by this compound. So accordingly, this there is no reported value for this one. So now you will choose these 2 compounds. Okay, so accordingly, or all the compounds you can choose. Or if you scroll this right side, other informations also you can get. So what are the other information? So unit. So always remember the unit should be in nanomolar. So that is most important thing. So this is IC50 value, and then a unit in nanomolar. Unit should be in nanomolars. Okay, so these all the things you have to check. And then other things you have to check is assay type. Here look at these labels. Okay, so assay descriptions. So all the 3 compounds are tested on PIM kinase inhibition. So the format is single protein format. Sometimes the format will be different, so in that way you can't select that particular compounds. Okay, so this everything should match. Okay, so that's how we have to screen out the compound, required compound. So yeah, so how you can download it? So to download this IC50 value, so there are options, 2 options. Okay, either in CSV format or TSV format you can download. You can click it TSV format. Okay, so you can download, or CSV format you can download. So it will show here, and then you can directly download it. It's that simple. Okay, so this CSV and TSV format will give you the IC50 value. Okay, but the structure, either you have to download one by one, correct? So but that is not possible, right? So it's possible, but like it's time consuming, right? Whole day you have to do the same things to collect only 50 compounds also, which is like quite lengthy. Okay, so here it shows the download file. So you can click and then you can download the TSP file. So now structure. So we need the structure also, right? So how you can download these all three structure at once. Okay, so for that reason, you have to go here, related compounds. Okay, so just click it, related compound. Yes, so how many compound it is showing? 3 compounds it shows. And if you scroll down, so it shows in this way, 1, 2, 3. Okay, so select it, select all. Now you have options like SDF format. So simple dimension file format is there. Okay, so now you can click this SDF format and then you can download those structure in SDF format and then use to for QSR development. Here it will show. This is how we can collect the data. Okay, so this is data set collections. Yeah, I think pretty much uh, everything is covered. How to download. Yeah, from here you can click the file, you can download it. Okay, yeah. If there are any questions how to collect the data, I can answers. Okay, so if any other approach is there, so if you want to share, you can share. Otherwise, like I will go to next steps.
Sir, I have one question. So, what is the maximum number of compounds that we can download like this?
Okay, so there is no maximum number, but in QSR model, there are minimum numbers. Okay, so minimum. So this is bare minimum of 10 compounds are required, which is not good, but still, okay. So minimum of 10 compounds training sets required. But I never suggest to take a 10 compounds. So my suggestion is always go above 30 compounds. Okay, so then you can develop the good QSR model. So 30 compounds you can do. And then for downloading the things, so you can download 1,000 compound also, but the provided conditions is those 1,000 compounds should be in that particular assay format. So in our assay format, only 3 compounds were there. So that's why we were able to download those 3 compounds. Yeah. So it's simple. So just you have to go to IC50, click the things, do download. Yeah, after downloading, you have to prepare the file that I will teach tomorrow, because in today's software that is not required. Tomorrow's that is required. For today's software, like nothing is required. It's very simple. Okay. Yeah. So if everyone got it, like how to download. So now I will show you. From PubChem also you can do. Okay, so let's say I think this is the easiest one. Everyone are using the PubChem. So from PubChem also you can get the biodata. So from here, let's say the same thing. If you type here PIM kinase inhibition. Okay, so search it. So or any other terms, whatever you want to search. Okay, so below that you will get this bioassay. Correct. So you just click to bioassay. Yes. So many experimental value have been performed and published and publicly available. Okay, so this means so anyone you can click. So you can click on the basis of tested substance. So they have 5 compounds. Okay, so how much you need, you can click on the basis of that one, or you can choose it from relevance. Okay, so here like bioassay ID increasing, bioassay type A2 Z. Okay, so deposit oldest, modest. So you can like tested substance count. Okay, so let's say the tested substance count. So we need tested substance count decreasing. Let's say the higher to lower, so that like you can get it. This one. So if you click here, so how many compounds are there? They tested 168 compounds. Out of 168, all the compounds are active. Okay, so let's click here. But this is US patent, right? So I generally don't suggest to use the patent things, but for demo purpose, okay? So I'm just showing, but my suggestions is don't use the patent. Yeah, because I don't have an idea related. It's like how patent, either it's like either we can use it or not. So here you go to the data tables. Okay, so after that, all the things you can download it here. So here they have given IC50 value, PubChem standard value, that value it is given, right? So along with the structure, so here download options is there. So you can choose the CSV file format, data table actives, okay, all CSV, okay, in zip format. So anything you can download. And then for structure, you can download like one by one, or in that way you can download it. So this is another way to extract the data. Okay, so yeah, PubChem also you can use. Okay, so is there any questions, please? Otherwise, like I'm going to finish it off by demonstrating this QSAR in software. I think no questions. So for QSAR and softwares, what is required then? Okay, so let me show the data required for QSAR and software. So here is the things. Okay, so QSAR and software only required. Okay, sorry, RA and QSAR software only require this data. It only require Ismail formula of your compound and biological activity of your compound. Okay, so this biological activity, either it's in IC50 value or in PIC50 value is fine. But for other software, this biological activity value should be converted into PIC50 value. IC50 value you can't use. That IC50 value need to be converted into PIC50 value. But for today's software, it's fine if it is in IC50 also or PIC50 also, because we have an option to convert IC50 in PIC50. So PIC50 is nothing but it's logarithms of the IC50 value. Okay, so tomorrow I will show how to do that one. So only these two things required. So whatever the procedure I told you, so from that procedure only I have made this file. So I downloaded those files and then I have taken the Ismail formula and their corresponding activity. So I made this file. Okay, so that's it. So the thing is, title should be Ismail's and then BA. If your title is different, the software is not going to recognize that. Okay, so that's we have to keep in our mind. That's it. So this data is required, which we already have. So now where is our software? This is the software. Yeah. So after downloading, you can get these types of panels. So this left side is control panel. So you make sure this all the checklist are there. Okay, so generate PDF reportings, and then this is the training windows. And if you click here, this is the predict windows. Okay, so first we will develop the QSAR models and validate the QSAR models under this training option. After validations, okay, so we can predict the biological activity of newly designed compounds through this predict windows. Okay, so yeah, under training windows there is options here, upload the training CSV file format. So it can it takes the CSV file formats. The thing should be columns, two columns, one Ismail's and one biological activity. Okay, so you browse the file here, you go to your the sample. So this is my samples. Okay, so done. So now this is uploaded. Okay, so sample 0.9 KB. Okay, so if you want to see the data how it is looking in this software, you can do the preview. So data Ismail's and then BA. Okay, so this is we uploaded the software, oh sorry, data. So now these are the things which we should understand. So what you want to do. So during theory sessions, I explain about the fingerprint region. So what type of fingerprint regions you want to use for your development. Okay, so here you have options. If you click Morgan fingerprint, okay, so and then hybrid fingerprint. So hybrid fingerprint include the Morgan fingerprint plus RD-kit physiochemicals, and it also include the topologicals. Or if you only need the RD kit, you can choose. So, but my suggestion is you go for the hybrid descriptor so that you can generate as much as descriptor required for the model development. Okay, so yeah, I'm choosing the hybrid one. So now splitting method. So I told you, like, we have taken the, how many compounds I think we have taken? I don't know how many we have taken. Not nine compounds. Yeah, 20 compounds we have taken. Okay. So 20 compounds means 20 sets we have taken. So these 20 sets we have to split, like into training set and test set. So how we can split? You have options here. Okay. Either you can choose the random methods, or you can choose the scaffold split methods. Okay. So it's up to you. So, but like I'm choosing the random one. So test fractions, as I told, like it's 70 into 30. Okay. Split, let it be uh, as it is, so no need to change. So I told you, like, no need to convert the IC50 into PIC50, because we have an option here in this software. So you can click if you have used the IC50 value. If you have used the PIC50 value, no need to check this. Okay, so I think mine is PIC50 value. It's not IC50, so no need to do that one. Here unit also you have to consider. Okay, so you have to be careful about the unit. Yeah. So now Morgan radius, let it be 2, and then fingerprint bits. So how many fingerprints it will generate? So 2048 total descriptors it is going to be generated. Okay. So top features like plot 30 plots, or you can choose any other things, but like I suggest, let it be, as it is default setting. So now variance filter. So this is very important. So what does it mean? So because it automatically does, it filters the variance descriptors. Okay. So like whatever the variables or the descriptors are matching to each other, or if any 0 is there, or like all the variables are ones, or like that. So many other parameters are there. So if you check this one, so it will automatically remove those things from the model development procedure. So another important thing is correlation filters. So generally this correlation threshold should be 0.95. That means if there is a correlation between two descriptors which is more than 95%, so it will automatically remove those descriptors from the analysis. That means the chances of correlations is going to be reduced. That means your model will be, good to fit. So it's a goodness of fit type of things in a statistical term, right? So it should be 0.95. Yeah, it's a standard as per this ORCD guideline. Yes. So now the algorithms, which I explained before. So here you have options. You can choose the algorithms as per your number of training set, as per your research methodology. Okay. So as per your uh, convenience, you can use the algorithms. You can also choose here. So random forest is there, extra tree is there. Okay. So east gradient boosting is there, raise is there. So many other things are also there. So you can choose it. So I'm choosing the random forest. So another thing. So now let the AI select the best model for you. Okay. So no need to manually find out and select. So if you click this one, enable AI automated selection. So AI will automatically select the best model for you. Okay. So these hyperparameters, I think no need to change. Okay, let it be as it is. And then now after that, like validation settings. Okay, so till here we are going to develop the models, okay, using the random forest. And then since we have enabled this AI auto model, so it will test with other things also and then give us the best result. And then now, after developing the QSAR models, so we have to validate that. So for validations, what all we need, that ORCD guideline as per. So you just check. So I told like randomizations is very important. So you check this one also. Okay, so how many permutations you need? This permutations means 20 times it will, find out the chance correlations in permutations, that one. So yeah, so you check all these parameters and let it be. And like reliability requires, so check it. And after that you just train the model. So it's that simple. Okay, in one go you can do. So you just upload the data and then select your parameters. Okay, and then till model development, use your algorithms, and after that use your validation parameters as per ORCD guidelines. Okay, and then just click train model. Yes, so it's running. It's running like featuring, splitting, pre-processing descriptors like training models. So everything is running. So you just wait it for a minute or some time too. So it is showing like we have uploaded 21. So there are no invalid data sets. So which is good, right? So yeah, so all the data sets are valid. So yeah, it will take a minute or two like that. Yeah. So if any questions by then, like I will be happy to answer.
Shankar sir, we can go ahead. No question.
I have one small question. So you showed us the Excel file, right? So based on what parameter you chose those drops?
Shankar sir, are you there?
Okay, so yeah, so it's valid questions, right? So am I audible to you?
Yeah.
Yeah, so it's valid questions like, so based on what parameters you choose those 11 compounds, right? So uh, for this 11 compounds, so sorry, 21 compounds or 20 compounds. So this is only for the demonstration purpose. I didn't use any of the criteria to select, but for your experiment, you have to use the criteria. Okay, so the criteria will be your IC50 value first. Okay, so like sometime you can use the better IC50 value. Suppose the IC50 value less than 100 nanomolars. Okay, so you have to select on the, suppose you have downloaded 500 of compounds from those database, and then now you don't want to use 500, so now you have to screen out. So how you will screen out? So you will first screen out on the basis of your IC50 value, because like better the IC50 value, better the model, correct? So another criteria is the structural similarities, or what is the core structure of those all 500 compounds. So you are going to check that. Okay, so how those structures are strongly related to each other. So in that way also you can select. Okay, so another way of the selecting those or screening out is actually second step. Okay, so you will calculate the descriptor. So from descriptors, if any of the descriptors are beyond the limit of ORCD guidelines, what does that mean? Let's say the some of the compounds are showing very much resemblance with each others, so no need to include those types of compound in our QSAR model set. So in that way, okay, so other parameters as per your research things. Okay, so as I already told you, like cell line based assay, the same assay should be uh, choose for all the training models. So these are the some criteria you have to follow. Obviously more criteria are there, so those all criteria are based on your research objectives. So these are the basic things. Yeah. Yeah, I think we already got the training compounds, right? So it's already got it. So here, so what AI is saying, so AI is like saying us Reed's model is the best models. So where is the Reed's model? So these fourth models. Okay, so if you see the Q-square CV value, so now the use of ORCD guideline is there. So now we have to go through our cutoff value and then compare with our obtained value, right? So this is 0.71. So actually above 0.7 is acceptable, but if it is near to 1 or near to 0.9, that is very good. So this is also 7. So this is root mean square error. So this is actually a good if the value is very less. If the value is 0.01, something like that, or the value is 0.1. So low value of root mean square error is better. Okay, so among these all five models, so AI is saying us this RIS models is best. So we can go with this RIS models. Okay, so where we can get the result? So if you go down, so here, okay, so you can find out here like predictions training test sets in CSV format or summary CSV or PDF reports. Okay, so if you click the PDF report, so it will generate the PDF report. So from where you can get it, so you just go here like open folder. For metric summary you have to click it, so it will give you the summary in CSV format. For predictions of training set and test sets, click it and go to here. So I have generated the PDF report, and also here is I check this things also, so I can easily get it. So you just click this open PDF folder. So if you click it, so you will go to this one directly. Okay, so now you just open this, so you can get all the results which you can use in your thesis for the publication. So here they have mentions everything, and now this all the value you have to verify with ORCD guidelines. Like for the training set, what is the criteria? Like R square is 0.99, which is very good. Okay, so R square for test is minus 1.00, which is not good at all. Okay, so like this, so external validations, these things is there. So everything's now you have to cross verify with ORCD guidelines. If these all the parameters are matching with ORCD guidelines, then you can select that model for uh, predictions. Okay, if it is not matching, some of the parameters are not matching the ORCD guidelines, what you have to do, we have to again go back to our software, and we have to change any one parameters, either the descriptor, you have to choose only one descriptors. Okay, so or you have to choose the algorithm change the algorithm these different algorithm models, or any of these parameters. So you have to do the hit and try. So it's like hit and trial type of things until you are getting the best models. Okay, so like this. So now you have to go through all these things. So because the test R square value is not good, so this is not that much reliable model, we can say. Okay, so and this is the linear curve, so which is required to include in the manuscript. Okay, and this was the linear that curve also, but this is like applicability domains. Okay, so this is standardizations like Y randomization. So this is Y randomizations. Okay, yeah, so this is Williams plot. This is applicability domains. Actually applicability domains is good, but like our training set is not good. Like training set, that R square value is not good, but applicability domains is all the compounds fall under the guidelines or under the this one. Okay, limit. So this dash line is the limit. So all the training set and test set, if they belongs inside this dash lines, that means our models is applicable. So this is external validation. So in external validations, it is fine, but somewhere the test models values, R square values are not good. So in that way we have to analyze. Okay, so let's say now this is the good models for the demonstrations only, I'm saying. Okay, so demonstrations part I'm just saying. Now let's do the predictions using the same models, right? So now what is predictions? Now we have to design the new compound. Okay, so design the new compound, so and then try to find out the PIC50 value of those compounds. So let's say I don't know what are the new compounds. I didn't generated the new compounds. Okay, let me take it from this one only. So let's say these are the sample predictions here, like some compounds names are there. Okay, let me take these new design compounds, let's say. So what are there in design compounds? Only Ismail formula and name is there. So in this way you have to keep. So name you have to keep, and then Ismail formula you have to keep. So these are the new design compounds. So now the developed models we will use to find out the IC50 value of these 100 compounds. It's very easy. So just you have to go to here, that prediction or predict, okay? And then after predictions, you have options here, or choose the current model. So which models is there? So you have to choose the current models. So it is automatically giving you the current models or hybrid models, okay? So let's say that we choose the hybrid model. So let's say the hybrid models, okay? So, and then you upload that file here, like browse the file, sample predictions, and you check these all the things so that it will calculate the things, so reliability and everything. Okay, so it's already uploaded and now predict it. So you can predict here. It's already predictions is there, like predicted biological activity, PA is given here. Okay, so this is your predicted IC50 value. Okay, actually it's a PIC50 value. Now PIC50 value you have to convert into IC50 value. So predicted IC50 value of all 100 compounds. So you can use it for your publications. If you need this data, you can download it in that CSV format here. So download it. So it can be downloaded in the result section. So it will go. Yes. So in this way we can use this software. Okay. So where you can get all these things, all the data is in this one. Okay, so you have to go to that QSAR, our RAN QSAR folder, open it, okay, and you have to go for the result. Okay, so and then in result, in report, you can get those all plots and all the things. Okay, so let's say, so these all plots you can get, which you can use for the publications. Okay, so and then like prediction value, you can get it from this prediction value. Okay, so like predicted result, if you open this Excel file, you can get it. So here, yeah, okay. So this is your predicted PIC50 value, which you can use. So now among these 100 compounds, so on the basis of this predicted value, you will find out which compound is best or which compound need to select for the synthesis and validate through experiment, because on the basis of this value you can use. So no need to synthesize all the 100 compounds. So in that way it saves the time and then money also. Okay, so that is the use of QSAR model development. Yes, in that way you have to do. So all the results you can get in that QSAR software inside folder. Okay, so you be sure about that. So this is my QSAR software folder, RAN QSAR, RAN QSAR, and you go to this one and go to the result. Okay, so all the result will be available here. In reports, you will get the reports. Okay, suppose this one, this training metrics report. Okay, this is another one. I think yes, today's this one only. So this one, yeah, this is the today experiment. So all the result will be available in that one. So these all the results I did previously. So but it's still available there. Okay. So and then predicted value, you can get it from the predictions. Okay. So these models is also there. So you can use those models in near future. You can open this and then you can again do the things. Yeah. So that's it. So this is all about the RA and QSAR software to predict the activity and develop the QSAR models. Also validations of QSAR models. It's very simple, very user friendly. They use the machine learning and AI to develop, so that like most of the manual things or the individual things, they bypass through it. No need to do, software automatically does. Okay, so in that way it is good, but sometimes we have to be very careful that the model is, you know, reliable or not, especially in ML based models. Sometimes models undergo into, you know, overfitting type of things. If your result is very good, that is also you have to cross check. Okay, so either the model is overfit or not. So that things we have to be very careful. And yes, finally, these all the things, all the values are predicted value. They are not confirmatory value. Okay, so you have to keep that things in our mind. Yeah, thank you.
Thank you so much, Shankar sir. There might be some question, but it's already too late. We will catch up from here tomorrow. If there is a brief any question in chat box, let me pull it down. Uh, Dil Kapoor is asking, hello sir, from where we will find the sample prediction data set for further prediction analysis?
Yeah, I think I already explained those samples.
Yeah.
From Campbell database you can do, and then from PubChem you can do. Yeah,
Okay.
you can pull out from there.
Yeah. Thank you, Shankar sir, for today's time, and we will continue from here tomorrow, same time. See you all tomorrow.
Yeah.
Do any have
So for
any?
tomorrow sessions, yeah. So for tomorrow session, please download that QSARN software. Okay, I think you can't download that one because you have to mail to the developer. So developer will
Yeah.
provide the file. Okay. So, but tomorrow we are going to learn that software, and which is like pretty much good, but like it's extensive. So many things we have to do manually. We have to write so many things, but that is the correct way of learning the QSR. So today's software was very easy. Okay, so like one click go type of things, but so many things are automatically done by the software. So it's quite impossible to understand what actually going into the development. So which we will learn tomorrow, okay, obviously. Yeah.
Yeah.
Thank you.
Thank you so much, sir. Thank you, everyone. Today, around 40 people we connected from US, some are from Korea, India, and mostly from the Nepal. Thank you, everyone. See you tomorrow. Now I close the meeting.